our next speaker, Robert Phair, who is with us here from Integrative Bioinformatics Incorporated, a local company. He's been collaborating with us sort of out of interest and the goodness of his heart for a couple of years. And he's about to present us with a theory for CFS that I think a lot of you already know about, judging from the questions that happened even before the symposium began. And we're looking forward to hearing the latest about that.
Well, Bob came to us, I think like 3 years ago, and said he had read something in the Stanford magazine, medical magazine, about chronic fatigue syndrome, and he said, I want to try to help. So he started a small company, Integrative Bioinformatics, here locally, and said, I'll donate one day a week of my time. So he started coming 1 day a week and getting involved, learning how to do sequence analysis and all sorts of things that he hadn't done before. But he was very, very willing to do all of that. Now, Robert is another nerd. That's not an insult, it's a compliment. From MIT, and as an engineer, and then went and learned human physiology. And so he knows both of those really well. And his goal was to be able to make predictions and integrate data in a kind of a systems engineering fashion. Now, that wasn't very popular with NIH and review committees, and so he decided, he was at Johns Hopkins, decided to quit and set up his own company and do it as a consulting. And that has worked out pretty well. I'm glad you did that. Anyway, this was a perfect problem for him, and he came to say he wanted to help, and I said, oh my gosh, I've been really, really wanting a systems engineer that knows human physiology, because I think that's where the answer is. And but there are almost none, because NIH won't fund them. And so it was fantastic that he came here, and I really loved the idea. He will admit that we don't know if it's right or not, but we're testing it. And he's going to tell you about the results of those tests.
Thanks, Ron. All right, so I want to ask, how many of you have read David Bell's book? This is called The Doctor's Guide to Chronic Fatigue Syndrome. Now that's a good number of hands. This is the best book on the subject. It was published in 1994. And I wish there was a supplement to this book to cover what's happened since. That supplement is being written by the scientists at this meeting. I've learned a heck of a lot from those people, and I'm glad I'm here. So, chronic fatigue syndrome is a complex disease. The human body is a nonlinear system. David Bell says in this book that if you want to treat chronic fatigue syndrome, you have to know the symptoms, and you have to know your physiology and your pharmacology. But if you want to cure chronic fatigue syndrome, you need to know its underlying mechanistic basis. So I want to talk about today is a new way of thinking about chronic fatigue syndrome that has helped me, at least, pull a lot of pieces together to see how the various symptoms and the various measurements we can make tell a systematic story. So to do that, I'm going to talk about these three topics: genomics, biochemistry, and nonlinear systems theory. Now you might be saying, what's nonlinear systems theory got to do with biology? And I've been asked this question throughout my career. As Ron alluded to, it's hard to get biologists to think about mathematics. It's also actually hard to get mathematicians to think about biology. But 45 years ago, I made a decision that my father never forgave me for. That decision was that I took my newly minted MIT electrical engineering degree and I went off to get a PhD in physiology. He thought this was a terrible idea. He's an engineer, my brother's an engineer, his dad was an engineer. I'm supposed to be an engineer too. And so I want to convince you not only that nonlinear systems analysis belongs in the study of biological problems, but that it can actually explain some of the things about CFS that have remained inexplicable. So, let's start where I started. I started with David Bell's book and with Osler's Web, Hillary Johnson's book. And there were a lot of stories in those books about CFS epidemics. The existence of these epidemics is, to me, an important feature of the disease. And the fact that you can have attack rates, that means the fraction of the people who are exposed to whatever it was, that is upwards of 10 or 25%, as it says in the slide, says to me that it's possible that any predisposing mutation is common. And so, as I arrived in the Stanford Genome Technology Center and started talking to Ron, it was just about the time that the big data study, the one funded by the OMF and run at Stanford, was coming to fruition. And so I had suddenly access to the genomic sequences, the genome sequences for 20 severely ill CFS patients. And so I started searching not for rare mutations, but for common ones, because of this feature about the presence of epidemics, like the one at Incline Village and the one at Lindenville, New York. And so I was especially interested in cases of mutations where the mutation might produce or uncover what's called a bistability. So I'm going to talk specifically about one such mutation or set of mutations today. But at first I need to define for you or illustrate for you what a bistability is. So here's a diagram. This diagram comes from a mathematician's paper. It's from a journal of physical chemistry. And it describes what's known as a bistability in a nonlinear system. That means that this system, and it can be the human body, everyone should know that the human body is incredibly nonlinear. Nonlinear system can obtain two different steady states. One of them I'll just use as an example, the one at the top, closest to the letter A in the diagram, is a solid line representing all the steady states that are healthy steady states. And the solid line at the bottom is a curved line, represents a whole set of unhealthy steady states. And the points, they're labeled with mathematicians' rigor, lambda 1 and lambda 2, are called the limit points. And the hypothesis that I want to test here is that CFS represents a collision with one of those limit points when you're on the healthy steady state. So in the upper left, I think I have a way to point to that. Right here, at that point, you fall off of the curve of healthy steady states and drop down onto this lower unhealthy disease steady state. The axes of this graph are not very clear. They are system parameter on the horizontal and system variable on the vertical. They represent features of the biological system that we're studying. And this presence of two steady states is the thing that we're looking for in those genomic sequences. Because we want to see something, a message to us in the genome that tells us that a bistability is possible. So what was the first such genome mutation that we found? That's here. In chronic fatigue syndrome, in these 20 severely ill patients, we found several mutations in one gene called IDO2. I'll tell you in a minute what IDO2 is. But you can see here that across the horizontal in the blue bar, there are some amino acid symbols followed by a number, followed by another amino acid symbol. These represent mutations. It's an arginine converted to a tryptophan in the first column, for example, in the 248th amino acid of this protein. And that mutation breaks the protein. The protein does not work if it has this mutation in it. Nevertheless, this mutation is very common. So if you look in the line, what did I do with that thing? If you look in the line that has the CFS allele frequency, that says that our 20 patients have that 55% of the time. And the 1,000 Genomes Project, that's one of the standard reference genomes, has it 42% of the time. So it's a very common mutation. 42% of the people in the room, maybe even more, since we're studying CFS, have this mutation. It's significant at the 0.03 level, not the 10 to the minus 9th level. So I can't say it's perfect, and I don't intend it to be. What's surprising is that even patients who don't have that very common mutation have another, or even one, or two, or three other mutations in the very same protein, IDO2. And although I don't know that all of those are broken biologically, I do know that all of the prediction software tools, one of them I've listed here is SIFT, that attempt to tell from the sequence whether or not it's broken or likely broken, predict that all of these are broken. Even the one in the middle here is a premature stop codon, so that's what we call a nonsense mutation. It doesn't even make the full protein. So these mutations provided a hint about what was going on in these severely ill patients that I wanted to follow up. Okay, so now what's IDO2? IDO2 is the first enzyme in a pathway known as the kynurenine pathway. This is the major degradation route for an amino acid called tryptophan. This is the least frequent amino acid in your proteins. Tryptophan is an essential amino acid. That means you cannot make it yourself. It is only available in the diet from organisms that can make it themselves. And so tryptophan has two fates that we're going to talk about. There are other small minor fates, but the big ones are the kynurenine pathway that goes from east to west in this diagram, east to west. And there are two enzymes that catalyze that first reaction. The IDO2 enzyme I was just telling you about with its mutations, and IDO1, which does exactly the same thing. And so you say, how can it even matter? This is what I was saying to myself. How can it even matter that IDO2 is broken when in fact IDO1 works just fine and does the same thing? So it took me weeks, months maybe, to figure this out. I don't know that I figured it out, but I've got one way that it might work. Okay, just to make the system complete, there's a pathway going from north to south here that also is important for the fate of tryptophan, and it is the production of serotonin, a molecule you've probably all heard of. It's involved in sleep regulation and many other things like controlling the immune system. So what could it be about the IDO system that actually matters, since IDO1 works fine and it's only IDO2 that's broken? So that answer came from enzyme kinetics. So this is one of the more complex slides I have. But, I've learned that the CFS patient community has read more papers on CFS than I have. And so I'm not worried that these slides are over the top of your heads. I'm worried that you're going to ask a question I can't answer. Okay, so on the left-hand side is a plot of tryptophan. That's the molecule that's the substrate for this enzyme, the molecule that's converted to other molecules. And on the vertical axis is the flux through that enzyme. That means how many molecules are converted to product in a unit time. So maybe 1,000 molecules per second, something like that. And on the horizontal axis is the concentration of the tryptophan that's going into that reaction. And you see for IDO2, the one that's broken, IDO2 has the classical shape. This is what we call a Michaelis-Menten curve, named after the two women who discovered this feature of enzymes back in the 1800s. And it has the right shape for almost all enzymes. But for IDO1, the shape is not the same. It doesn't saturate up here and become a maximal velocity. Instead, it goes over a peak and comes back down again. So at very high tryptophan concentrations, there's much less flux than there was before, much less production of kynurenine. And so instead of having the sum of the total flux be something like a Michaelis-Menten curve, if you get rid of IDO2, it looks like what you have on the right. Right? So IDO2 is in effect a backup system. It works at very high tryptophan concentrations to maintain a flux through the kynurenine pathway in the presence of a very high tryptophan concentration. So now you see what could go wrong. IDO2 doesn't work, it's broken, so now there's a much lesser flux. And as soon as you go over this peak, as soon as somehow, and I'm thinking this is what happens in that initial stressor that you've encountered that put you into the CFS disease, you were pushed over this peak, and once you get over the peak, now you're in trouble. Because now, every time you increase the tryptophan concentration, the flux through the enzyme goes down. And every time the flux through the enzyme goes down, the tryptophan concentration goes up. And every time the tryptophan concentration goes up, the flux through the enzyme goes down, and so on and so on. And you are pushed all the way down the curve. You're stuck on the other side of this peak. And as a result, you're stuck in what I call the IDO metabolic trap. So, key points so far. Severely ill CFS patients have on average 1.7 broken copies of IDO2. There are only two possibilities. 1.7 is a big number compared to 2. And without IDO2 as a backup for at high tryptophan concentrations, inhibition of IDO1 by its own substrate opens the door to a pathological steady state. This is an example of the bistability in nonlinear systems that I've been talking about since the beginning. So the next thing we did was show that that's not just hand waving. I stood there, I talked about the going over the peak in the IDO1 curve. Yeah, that sounds pretty good, but we built a mathematical model. This is actually the part of the project that I actually do for a living, so I'm happy with this. This is a model of that system that I drew, the tryptophan pathway through kynurenines and the tryptophan pathway to serotonin. And what the model shows is that a very large increase in tryptophan concentration, artificially produced in the model, for only 10 days, causes a change in tryptophan, as you might guess, inside the cell, that when you remove that high tryptophan in the extracellular space, actually returns to normal. But it takes 30 weeks, half a year. And if you do that longer, if you have the high tryptophan for, I think this one is 8 weeks, so 2 months worth of stressors, then you never return, even though you have removed the stressor. So I take this tryptophan right back to where it was in control. And everything in the cell stays where it was. So this is a very high level of tryptophan. This is a very low level of kynurenine, the downstream product. And this is the flux through that pathway, the IDO1 pathway. It's very depressed. So the model is consistent with the idea that not only is it fun to look at going over that peak, it actually happens in theory. So now, this model makes predictions that we can test. Now, I couldn't test these things myself. I'm a theorist now. I did a lot of experiments when I was at Johns Hopkins, but that was a long time ago. And furthermore, these experiments are a lot harder than the ones I used to do. So that meant I needed a team. And so fortunately, the OMF, when they funded this grant that I'm working on, also funded a grant for Ron's team. And so Julie Wilhelmy, Curt Fischer, and Sundari Suresh have made it possible to make the measurements I'm going to show you. Curt Fischer is an expert in mass spectrometry, and Julie Wilhelmy has the best experimental hands I have ever had the pleasure to work with. She's the best in the lab I have ever seen. So this team wanted to test these predictions. In affected cells, cellular tryptophan will be increased. This is the tryptophan level inside the cells. So it's not going to be outside. It's inside the cells. Cellular kynurenine and the kynurenine to tryptophan ratio will be decreased. That's another prediction. And finally, the flux catalyzed by IDO should be lessened in CFS. So we undertook to do that. We've only been doing this for 8 months, so this is only n equals 6 patients. And on these two figures, what we have is the kynurenine concentration in molecules per cell. For the red data points are the healthy controls, and the blue squares are the CFS patients. And you can see that kynurenine is, as predicted, substantially lower than it is in the healthy controls. Time equals 0 here is when we added a metabolic tracer. I didn't mention that, I should have in the design. Tracers are here used to follow the fluxes. So I'll show you in a next slide or two where the tracer goes. But this is what's actually plotted on the horizontal axis is the time after tracer addition. Over on the right-hand panel, you see that the tryptophan in the cells is also high in the CFS patients as predicted. That's not statistically significant. I'm hoping it will be by the time we get to n much greater than 6. So the last one then is that the cellular kynurenine to tryptophan ratio is decreased as predicted. This is highly significant in n equals 6 patients. So those are all consistent with the theory. And we also measured that flux that I told you about using the carbon-13 labeled tryptophan. You can see that the blue curve here for CFS is again much depressed compared to the production of kynurenine in healthy controls. So my main points for each of those 3 topics that I discussed are at least some predisposing mutations are common. We have data on a particular set of mutations in IDO2. In the biochemistry part, the blue circle of my Venn diagram, there's tryptophan pathways to kynurenine and to serotonin. I showed you what those were. And then showed you about substrate inhibition, a particular feature of IDO1 that makes it relevant to our bistability that might be the characteristic of chronic fatigue syndrome. And then we did the simulation to show that it works in theory, and that some of the predictions of that model are confirmed experimentally. So this is important for CFS because metabolic traps explain MECFS as a jump from a physiological steady state to a pathological one. This jump is made possible by predisposing common mutations. It's initiated by a relatively improbable combination of triggers. They're not always the same triggers, just any trigger that allows you to get into the bistable state, the low state of your bistability. And finally, it's sustained not by something acutely broken, but by a biological defect that evolution hasn't solved yet. So, the ideal metabolic trap is one example of this kind of bistability. It has some genomic and computer simulation support, and it has makes predictions that have been confirmed experimentally. So I want to just spend a minute on why this might matter to you. This is a diagram of the brain in cross-section, and in the purple, you see 6 nuclei. These are called the Raphe nuclei, and they are the neurons of the serotonergic nervous system in the brain. And those neurons are capable of controlling a vast number of portions of the brain that are relevant to CFS. I'm being shepherded off the stage, so let me tell you that cognitive function, movement, smell, dysautonomia, motor neuron dysfunction, dopamine system, all of these, headache, air hunger, POTS, parasympathetic dysfunction, and pain, all of them have inputs from the serotonergic nervous system in the brain. And I think that's relevant to the disease. How will we proceed? We're going to pursue the trap intensively. We're going to initiate collaborations. I must say, in this week, probably 4 new collaborations on the trap have emerged from our scientific discussions. And we're going to design and test therapies based on the trap and see if we can make them work. If, and that's a big if, CFS is caused by a metabolic trap, then our job is to get the patient from the sick set of steady states back up through the limit point and onto the domain of healthy steady states. Thank you.